Greetings, everyone, and welcome to the first of our IAFNS webinar series with the Beltsville Human Nutrition Research Center of USDA ARS. We're thrilled that so many of you are joining us today to talk about sources and intake of added sugars in the U.S. diet. My name is Gwendolyn Jones, and I have the privilege of serving as the Executive Director here at IAFNS. IAFNS, or the Institute for the Advancement of Food and Nutrition Sciences, mobilizes our membership from government, industry, and academia to drive, fund, and lead actionable science, all to advance public health. We are focused on catalyzing the creation of scientific knowledge by funding researchers and convening experts. Our collaborative and inclusive structure empowers members of the entire food and beverage ecosystem to bring forward their diverse perspectives. Everything we do as IAFNS is grounded on our 4 core values: scientific integrity, transparency, collaboration, and public benefit. Today's webinar is approved for continuing professional education credits by the Commission on Dietetic Registration. And I want to note that a link will be shared with participants after this meeting so that you can go claim CEU credits and a certificate later. As we get going and finish out my welcoming remarks here for this webinar, I want to highlight how to use our GoToWebinar system. First, you'll notice in the top of that participant window is an orange arrow. That orange arrow allows you to expand and contract the size of that participant's window. Second, and this is perhaps the most important point, there's an opportunity for you to engage directly by typing in questions and comments in that question box. Questions and comments that are typed are only visible to the IAFNS staff and not to other attendees. And following the presentations today, the panel will be available to answer questions that you may have. So with that, it is my pleasure to kick this whole thing off by introducing Dr. Naomi Fukugawa. Dr. Fukugawa serves as the Center Director for the Beltsville Human Nutrition Research Center. And additionally, we're thrilled to note that she serves as an advisor for IAFNS Sodium in Food and Health Implications Committee. So with that, Naomi, please join us on screen. There you are. Fantastic.
Thank you very much, Dr. Jones. And we appreciate this opportunity to share the research and highlight some of the research being conducted by BHNRC scientists in this webinar series. This next slide shows the advisory committees on which I serve. And I should mention, as you have mentioned earlier, the ARS, or Agricultural Research Service, is the intramural research agency of the USDA. As shown on the next slide, ARS does it all. It supports research that takes one from the bench to the field, to the plate, and to the impact on health, and then back again. The BHNRC, or Beltsville Human Nutrition Research Center, as shown in the next slide, is one of 6 human nutrition research centers in the USDA ARS. BHNRC is the oldest and the largest and located on approximately 6,500 acres of land between Washington, D.C. and Baltimore, Maryland. And it includes crop fields, animal housing, as well as laboratories for all of the scientists. HNRCs conduct intramural research and supports collaborative research with academia and industry focused on food, its components, and the impact on wellness of the population. There are 3 fully federal HNRCs and 3 cooperative centers, which include Baylor University, Tufts University, and the University of Arkansas. On the next slide, one of the unique responsibilities of ARS and the BHNRC is the monitoring of food composition, which has done since Atwater for over 100 years. After more than a decade of planning, a modernized version of the USDA food composition data system was released as Food Data Central, as shown on the next slide. It consists of 5 distinct data types, each with its own unique purpose. And you'll hear more about it later today. On the next slide, we hope you have registered or are considering registering for the complete series of webinars highlighting BHNRC's research. Focusing on today, which is shown on the next slide, we will look at the impact of sugars on health and wellness. So I'd like to turn it over to you to begin with the first speaker. Thank you very much.
Absolutely. Thank you so much, Dr. Fukugawa. And with that, I'd like to invite Dr. Harry Dawson to join us. There we are. Fantastic. Good to see you, Dr. Dawson. And we're going to turn presenting rights over to you and ask you to talk about comparative genomics and the sweet taste of perception. So please, let's have you pull that out to presenter mode and we'll be good.
Well, good afternoon. I work at the Diagenomics and Immunology Laboratory, and the work I'm going to present today is a small part of what we do. The other 2 components of my research will be covered in sessions 2 and 4. We're going to make a disclosure that I work for the USDA and I have no external financial arrangements. So we've been publishing in comparative genomics for about 10 years now, and we've got about 8 papers, 9 if you count the one that's in the works now. And we ask basically 4 questions. Is the gene there or not? And that seems like a relatively easy question, but it's actually very difficult to establish whether or not the different species have the gene. We also care about whether or not the gene is protein, or the protein is structurally and functionally similar. We care about tissue expression, and we care about whether or not the gene's regulated in a similar manner to stimulus. So in the past, we've focused our attention on immune response genes, and we've turned our attention to the so-called nutriome. In this data here, it's under construction. We have some more work to do, but when we compared 1,500 genes that were involved in nutrition and metabolism, we found that in fact, hold on a second, there you go, laser pointer on, that there were a large number of unique genes among the 3 different species that we use. And there's a core group of genes, which are about 55%. So if you take a look at here, there's 209, 240, and 66. Pigs have about 4-fold less unique genes than the other 2 species. But if you look at the genes that are conserved between pigs and humans, you see there's relatively a 10-to-1 advantage of using the pig as a model. And that's how we view it. We start with the mouse, we move things into the pigs whenever we can, and we would like to study them in the human if we find conserved pathways. So focusing our attention to today's presentation looking at taste receptors, and we published a little bit of about this several years ago. If you just count up the numbers, you can see that they're relatively comparable for everything other than the bitter taste receptors. In humans, there's about 25. In mice, there's about 50% more. In pigs, there's about 50% less. But that just tells us the number of genes. When you take a look at orthology and break it down, mice and pigs have one-to-one orthology for the sweet and the umami taste receptors, almost a complete conservation among the salty taste receptors and sour taste receptors. But if you come up to the bitter taste receptors, in fact, even though the mouse has more than the pig, the one-to-one orthology in humans, at least from a structural standpoint, there are 3-to-1 advantage here. So again, the pigs are somewhere in the middle. This is a 3D representation of the human sweet taste receptor as rendered by the AlphaFold artificially intelligence enhanced program. You'll see that it is a homodimer. There is a taste 1 receptor 2 and taste 1 receptor 3. You see there are 3 domains in both. This top part here is the extracellular region. It binds various sugars in a domain called a venous flytrap domain. And you can see some artificial sweeteners here also bind that site. The taste receptor group 1 receptor 3 also binds glucose and sucrose here. There are some species difference in this, in that the taste receptor 1 receptor 2 gene in fact is lost in cats and birds and some other species, dolphins being among them. And taste receptor 1 type 3, that is also lost in dolphins. And it's a common feature of cetaceans, like dolphins, to have lost taste receptors. And it's thought that they swallow their prey whole. They don't need to really taste, and they sense everything by their olfactory system. We know that there are some species-dependent differences in the ability to recognize, for example, aspartame, in that pigs and mice don't recognize it as sweet, whereas humans do. If you take a look at that venous flytrap domain comparison and look at the different species that we have here, you can see there is a nice segregation among the different types of species. The mouse, which is, in my favorite term, it's an opportunistic omnivore, which I consider myself to be also. Flying fox, which is a fruit bat. The pig, which is a true omnivore. The tarsier, which is a carnivorous primate. The baboon, which is another opportunistic omnivore. And then you have the great apes broken down into whether they're leaf eating or whether they're leaf eating and fruit eating. And the humans fall right in the middle of that. And humans are true omnivores. So if you just do this gross domain comparison, you can see that at a macro level that in fact there's not a whole lot of information we can glean from this, that doesn't appear to be any relationship between this at the raw homology level. So if you take the pig and the human and the mouse structure of this, again using this AlphaFold program and overlay, you can see there's conservation among all the major domains here. There are some minor differences, but these are regions that don't recognize ligand. Now, what do we know about the variations in this gene in humans, and particularly its relationship to sugar intake? Well, if you take a look at the literature, it's been described as a highly polymorphic gene. It's in the top 5% of all human genes. When I checked the human variant database this morning, there were about 5,200 entries. And if you're doing something like precision or personalized nutrition, this gene would be an attractive target if we could nail down exactly what relationship it has with sugar intake. So the literature suggests that polymorphisms in this gene are associated with sweetness sensation and sugar consumption. And that's a rough review of all the polymorphisms, but we were particularly interested in one where in position 191 of the human gene, the amino acid isoleucine is replaced with a valine. These are branched chain amino acids. It results in a small change in size. It's not something where we have a branched chain amino acid being substituted with a highly charged amino acid. It's a minor allele. It's an average of about 30% in the population, with variation ranging from 40% in Africans and 20% in Asians. It's been recently characterized as a partial loss of function mutation in that it doesn't go to the membrane as easily as the native form, but the ligand binding region doesn't appear to be affected. So this polymorphism in human has been associated with a lower intake of sugar in children, either a higher or lower intake in adults. And in this study, it was segregated among adults with a BMI greater than 25. And recently, it's been associated with reduced hemoglobin A1C levels. So if you take a look at the same species that we mapped out on the gross domain level and take a look at the flanking region for amino acids on 191, you can see, like the original diagram, it doesn't appear to correlate with what the animals eat. So at the top here, we have fruit bats with an isoleucine here, along with the mouse and pig. The tarsier, which is a carnivorous primate, has a valine there, as do the great apes and humans with this polymorphism. So this doesn't appear to indicate that there's some value in predicting whether or not this changes the structure of the protein. But if you want to formally evaluate this, we ran the same proteins through the AlphaFold program and overlaid them. This is the human taste receptor type 2, and this is the polymorphic version. And the arrow indicates where, in fact, in the protein that the structure would be located. And you can see there's a perfect overlay of the human and the variant. And if you do the same thing with our 3 species that we're interested in, you can see again that doesn't change the structure of the protein. So we move on to the third question that we ask is the gene expressed in similar cell types and tissues? Those taste receptors are not exclusively found in the mouth. As a matter of fact, they're widely distributed among the body, and we don't really understand what they're doing in other tissues except for in the gut. And this is a diagram that illustrates the TACE1 receptor type 2 and 3 in the gut. This is what's called an enteroendocrine cell. It senses glucose, and the signaling cascade results in the release of this glucagon-like peptide 2, which travels to the enteric neurons it sensed. It signals back to the enterocyte with the ultimate result of upregulation of glucose receptors at both the luminal and the side that adjoins the blood system. So the net effect is that its glucose is sensed and it's taken up and transported into the body. There's not a whole lot of good data that's comparative among the species, and I've limited it to just 2 species here. So if you direct your attention to the top panel, which is A, this is messenger RNA taken from the RNA atlas. The expression of this taste receptor 1 type 2 in the duodenum is high in humans, and it's actually expressed, but you can't really see it because it's at a very low level. But look over here at tissues like the testes, the liver, and the skin, and it's expressed in far more abundance than it is in the gut. And if you look over at the taste receptor 1 type 3, you can see again its expression levels are much higher than in the gut. But at least in that respect, the pig and the human seem more comparable. So there are papers in the literature, and this paper used immunofluorescence to detect TACE1 receptor type 2 in the gut of pigs and TACE1 receptor type 3 in the gut of pigs. And if you overlay the green dot with the red dot, you'll see that there are cells that express both and cells that express just one. And this is, in fact, if you do additional histology, this is an authentic enteroendocrine cell in the gut. So at least from that standpoint, we know that that is conserved among the species. Now, we can't consider sweet without other sensations. In fact, this top line here was taken from the Mary Poppins movie, and what it referred to was the fact that the Sabin vaccine had to be administered on the sugar cube because kids found it so horribly bitter that they wouldn't take it. And if you've ever worked in a restaurant or a kitchen, you know you can mask a lot of bitter taste with sugar. And you can also add bitter compounds to things that are overly sweet to bring down the sugar level. And there are many, many, many bitter taste receptors as we'd seen before, but this one in particular is of interest, this taste receptor type 2, member 38. The polymorphisms in this taste receptor have been associated with the perceived sweetness of sugars, energy intake from sweet tasting foods, sugar intake, and there is a paper in pigs suggesting that at least it's associated with back fat thickness in pigs. So this is a clade showing the evolutionary relationship of all the bitter taste receptors. I don't want you to try to read the individual ones because it's difficult. There are so many of them. What I want you to pay attention to are the general patterns, and there are 5 of them, where the human and the pig and the mouse have gone from an evolutionary standpoint. For the first 3, they're pretty comparable, but mice have gone off on their own down here relatively late in evolution, and then finally humans have really gone off the end down here in as far as evolutionary time. And we saw this before. There are 25 human, 36 mouse, and 18 pigs. When we take a look at the end of these nodes, if they're equal, we consider that to be an ortholog. And if it reaches the end of the node together, it's a 1-to-1 ortholog. And if you view that number here, again, we come up with 4 direct orthologs between mouse and humans, and then we have 12 that are pigs. But this just tells us whether the protein is there or not. It doesn't tell us anything about whether it's functional. So if you take a look at the different ligands that have been tested over the years, and this work has been done to a large degree down in Philadelphia by the Monell Chemical Synthesis Institute, you can see these receptors have various ligands that are associated with various compounds. Sometimes you have single ligands for a single receptor. Sometimes you have more than one that bind to a receptor, and then you have promiscuous ones like caffeine binding to receptors. And interesting, it's not always the same receptor that's recognized. But if you add up the number of 1-to-1 ortholog from a potential functional standpoint, pigs have 7, and mice probably don't have any direct orthologs. There's one here that may be an ortholog, but in fact, this receptor does not recognize this phenylthiocarbamide. This gene also helps predict whether or not your children will eat cruciferous vegetables. And this compound was used on filter papers in psychology classes to detect whether or not you were a taster or a non-taster. So it's been around for a while, and it partially predicts sugar sweetness. And if you can see, there's 1-to-1 orthology for a lot more pigs than there are on mice. But wait, if you take a look at a literature observation here, pigs are not as sensitive to quinine as humans. And our explanation we're looking at, do they express a different or reduced set of bitter taste receptors for quinine? And in fact, if you take a look at those, they do. If you break it down just like we did before in color and go to the end of the nodes, you'll see that humans have 9, mouse have 10, and pigs have perhaps 4. And what I really want to emphasize is something like hops, which is the component that gives beer its savory and bitter taste. You can see that there are various receptors for hops compounds, and in fact, humans and mice have about 4. They're not the same. Pigs have perhaps 1. We'd have to clone that receptor out and test whether or not. And the tragedy of it is that this poor guy doesn't even realize he's drinking beer. So with that, I leave you with the conclusion slides based on what I showed you and a lot more work that we've done mapping different regions. This pig taste 1 receptor type 2, the structure is closer to that of humans than mice. The presence of the isoleucine or valine in the 191 position of the human taser type 1 receptor 2 gene does not change its gross structure or correlate with any dietary pattern in other mammals. Mice have more and pigs have less bitter taste receptors compared to humans, but when you break it down into true orthology, pigs have 3 times more 1-to-1 bitter taste receptors than mice, but lack all of them that respond to quinine and bitter compounds in other foods like hops. So with that, I'd like to acknowledge my partner in the genome business, Dr. Celine Chen. I mentioned to you that we were working on a nutrigenomics project. That project encompasses 9 other labs. I haven't shown their contributions to that, but it'll be part of the larger project. And I want to leave you with the quote, remember that all models are wrong. The practical question is how wrong do they have to be to not be useful? So with that, I will take your questions at the end, and I'll turn over the presentation to the moderator.
Great. Thanks so much, Dr. Dawson. Let's next welcome Donna Rhodes. Donna, join us on camera and we'll transfer presenting rights to you. And while that's going on, I'll do a brief introduction and note that Donna is a nutritionist with the Food Surveys Research Group of USDA, and she's going to talk with us about how intake can be tracked using the What We Eat in America survey. So, Donna, please.
Thank you. And are my slides up?
Everything looks great. Thank you.
Okay, good. It's hard to tell from here, but thank you for that introduction. And I want to thank IAFNS for coordinating this webcam. I don't have anything to disclose, no conflicts of interest, except that I work at USDA and I do have a sweet tooth and love to eat sweets. Now today I will give a brief overview of what we eat in America and then highlight trends in sugar intake in the US. But my main focus today will be highlighting the Food Surveys Research Group website, both our databases and summarized data tables. NHANES is a nationally representative cross-sectional survey designed to assess the health and nutritional status of the civilian, non-institutionalized U.S. population. It's conducted by CDC's National Center for Health Statistics, and it's a continuous survey with data released every 2 years. The Food Surveys Research Group of the Beltsville Human Nutrition Research Center has lead responsibility for the dietary intake component called What We Eat in America. We collect 2 24-hour recalls on all days of the week. Detailed data are collected on food and beverages consumed, as well as time and eating occasion, where the food was obtained, etc. Now, a few points on the source of the sugar values that I'll present later. Total sugars includes both added and naturally occurring sugars. Naturally occurring sugars are found in milk, such as lactose, and fruit, fructose. The value for total sugars comes from USDA's Food and Nutrient Database for Dietary Studies, or FNDDS, and it represents mono and disaccharides. Added sugars include sugars that are added during processing, foods packaged as sweeteners such as table sugar, sugars from honeys or syrup, and sugars from concentrated fruit juices. These values are estimates based on a computation. An estimate is determined for the amount of added sugar from total sugar in the food, and then it's computed to teaspoon equivalents of added sugar based on 1 teaspoon equivalent being 4.2 grams of sugar. And these values come from the Food Patterns Equivalence database. I'll go into more detail about these later in my presentation, but first let's look at the added sugars for selected foods and beverages. As you can see, a glass of milk contains lactose, so therefore it has total sugars, but it doesn't have any added sugars like you would find in chocolate milk. Cookies and candy bars have added sugar. And you can see that a hot fudge sundae and a 12-ounce can of soda have almost 9 teaspoon equivalents of sugar. Now, values for all beverages and foods reported in What We Eat in America can be found in FNDDS or FPED on the link at the bottom of the slide. Values for added sugars can also be found in the USDA Global Branded Foods Database that Kyle will be discussing later during this session. Now let's turn to intakes based on dietary recalls in What We Eat in America for individuals 2 years and older. The intake of total sugars will be in blue and added sugars in green. As you can see, overall total sugar has decreased from 2003, when it was 133 grams, to 108 grams in 2017 to 18. Likewise, the intake of added sugars has also declined from 21 teaspoon equivalents in 3 and 4 to just under 17 teaspoon equivalents in 17-18. Now, this graph shows the contribution of sugar intake to daily energy intake for 2 survey periods. 2007 to 8 is on the left, and 2017 to 18 is on the right. Also listed are the mean daily energy intakes. And as you can see, there's been very little change between the 2 periods in total energy intake. But there has been a significant decrease in total sugar in 2017 compared to 2007 to 8. It declined from 24% to 21% of total energy. And that's represented by the reduction in sugar intake of 12 grams between the 2 periods. Now, there is no recommendation for intake of total sugars, but the Dietary Guidelines for Americans recommends limiting calories from added sugars to less than 10% of total calories per day. FDA includes added sugars on the Nutrition Facts Panel label to help us know how much added sugar is in a product. Now, to look at, are Americans meeting the recommendation to limit added sugars to 10% of daily energy? Now, while we have seen a decrease, the average intake of the population still exceeds the Dietary Guidelines goal of less than 10% of daily energy intake. In 2017 to 18, intake of added sugars was 13% of our total daily energy. But consistent with the decline in total sugar, there was a significant decrease in added sugars between these 2 time periods. Now, as I mentioned earlier, values for total sugars come from FNDDS, and added sugars are estimated in FPED. Both of these databases are available for download on the Food Surveys Research Group. The blue bar by the first star will take you to FNDDS, and the second star by the blue bar by FPED will take you to values for added sugars. Now, to spend a little bit of time, I want to highlight the difference between FNDDS and FPED. The FNDDS is an application database created for analyzing dietary intakes in What We Eat in America. It converts the food and beverage portions reported by participants into gram amounts and determines their nutrient values. A new version of FNDDS is released for every 2-year cycle of NHANES. It contains approximately 8,000 food codes, along with descriptions and a number of portion weights for each food code. Each food code has a complete nutrient profile for energy and 64 nutrients. One of those nutrients is total sugars. Now, the underlying basis for the nutrient values is Food Data Central, as was mentioned by Dr. Fukugawa at the beginning of this webinar. Now, FPED allows comparison of intake to the dietary guidelines for Americans. It disaggregates each FNDDS code into 37 food pattern components as defined by the dietary guidelines. One such component is added sugars, and here estimates are provided in teaspoon equivalents. Now, FNDDS and FPED provide different types of data as described, as illustrated here. From FNDDS, a hot fudge sundae with a portion weight of 180 grams has 437 calories, almost 200 milligrams of calcium, and 43 grams of total sugars. Now, this includes both naturally occurring sugar and added sugar. On the other hand, FPED disaggregates the hot fudge sundae into components. 1/3 cup equivalents of total dairy, solid fats, plus 8.8 teaspoon equivalents of added sugars. Now, in addition to the two databases that I talked about, the FSRG website provides what I consider a hidden treasure, our data tables and data briefs. Now, this treasure chest of summarized dietary intake data are a valuable resource, and there's no analysis required. I'll highlight the two different sets of data tables from our website, plus data briefs. Now, if you click on data tables on the yellow What We Eat in America bar on our website, there are 56 tables for each release of What We Eat in America in Hanes. Each of these set has data on total sugars as shown here with the blue bar. And these are provided for 27 age and gender groups. Other tables provide data by race, ethnicity, and income. Whoops. Now, another set of tables on our website provide intake data for the food patterns components, one of which is added sugars. Now, I took the mean and standard error directly from table 1G from our website to produce the graph showing added sugar intake among US children. So you can see that in 2017 to 18, the average child in the US was consuming 16 teaspoons of added sugars, and males between the age of 12 to 19 were consuming 21 teaspoon equivalents of added sugars per day. Now, this is added sugar intake for children. Kelly will be presenting detailed results for added sugar intake among US adults. I also want to highlight just one of our 48 dietary data briefs that are available on our website. These briefs summarize key results from What We Eat in America in Hanes. Brief number 32 details beverage choices among children. Now, the figure on the right shows the percent of children who consumed beverages and that by type. And note that nearly half of very young children, 2 to 5 years of age, reported a sweetened beverage on a given day. Now, a resource useful for analyzing foods and beverages, such as these sweetened beverages, is the What We Eat in America food categories, also available on our website. The categories group foods and beverages reported in What We Eat in America into about 165 unique categories, which can be combined into groupings as needed to address specific research questions. This table shows the groups and the main groups and subgroups. And as an example, if you look at sweetened beverages, it contains 5 individual food categories: soft drinks, fruit drinks, sport and energy drinks, nutritional beverages, and smoothies. So let's take a quick look at trends in soft drink intake over time. This figure shows the percentage of individuals reporting a soft drink during their 24-hour recall. The green line represents adolescents. The red line is children 6 to 11 years, and the blue line is adults. So from 2003 to 4 to 17-18, there was a dramatic decrease in the percent reporting soft drinks on a given day. Now, this is reporting any amount of soft drink, even if it was just an ounce or two. And the largest decline was seen in children 6 to 11 years. 58% reported a soft drink in 2003 and 4, compared to 27% in 2017 to 18. And this, it does include anyone that reports any amount of soft drink. But if you look at the amount that was reported, we have also seen a decline in that. So mean daily intakes of soft drink reporters has decreased for each of these age categories. 6 to 11-year-olds reported an average of 16 ounces when they consumed soft drinks in 3 and 4. And in 17-18, it was down to 11 ounces. Bigger declines were seen in the adolescent age group. I want to point out just another group of food categories important when analyzing intake of sugars. And this subgrouping includes sugars and jams, syrups, and toppings. Now, we don't generally eat a teaspoon of sugar unless you like Mary Poppins, as Harry pointed out. But most of the sugar, reported as sugars, is added to cereals, and really most is added to beverages. So when analyzing sources of added sugars, it's easy to overlook this addition of sugar that people add to their coffee or tea, or a topping added to ice cream. I presented values for a hot fudge sundae as a single code, reported as a hot fudge sundae. Now, another individual in NHANES may report a bowl of ice cream, and then they added some hot fudge to it. So this would have two codes, ice cream and hot fudge. But both of these individuals consumed ice cream and hot fudge. So it's important to include these additions added to foods, since they represent how we consume foods and beverages. I drink coffee, but I add flavored creamer to it. So I'm really getting 10 grams of added sugar in my morning coffee, which won't be counted if you just looked at my consumption of coffee. Kelly included these additions in her analysis when she looked at sources of added sugar, and she'll be discussing that. Now, in closing, I just want to acknowledge the support of Joe Goldman, a statistician in our lab, and Alana Mashve, research leader. And I hope I have increased your understanding of the USDA databases, FNDDS, FPED, and the What We Eat in America food categories. And I want to just leave you with a sweet note and report that on a given day, 1 in 4 Americans consumed candy. This is from 17-18 data from What We Eat in America. Now, NHANES does not report data by month, so I'm sure that consumption is a lot higher, will be a lot higher next week as we all eat all of our leftover Halloween candy. So thank you, and I'll take questions at the end of the presentation.
Great, thanks so much, Donna. Really appreciate that, and I know, and I can already see questions populating in. So please do be typing in your questions, and we've got Kelly Higgins joining us next. Dr. Kelly Higgins is a nutrition scientist at the Food Components and Health Laboratory at Beltsville, and she's going to share with us about where sugar appears in the diet. So, Kelly, please.
Great, thank you very much for that introduction. Just to confirm, can you see my slides?
So what we're seeing is the speaker view. If you could swap views, that's all you have to do in that. There we go. I think we're good to go.
All right, great. Thank you very much. And thank you, Donna, for that excellent presentation, which will be a great segue into the next component of our webinar series. I'm Kelly Higgins. I'm a research nutritionist in the Food Components and Health Laboratory. Today, I want to present some preliminary research we have done looking at sources and intake of added sugars in the US diet, particularly among adults, which was a project that we did in collaboration between the Food Components and Health Lab and Food Surveys Research Group. These are my disclosures. As Donna discussed, the US population is consuming added sugars at levels in excess of recommendations. Numerous governmental and health organizations recommend reduced intake of added sugars due to the health risks associated with excess intake, including risk of diabetes, obesity, cardiovascular disease, and dental caries. The Dietary Guidelines for Americans recommends consuming levels of added sugar less than 10% of total energy. But as part of the Dietary Guidelines process, the Dietary Guidelines Advisory Committee further recommends reducing intake of added sugars to less than 6% of energy to allow individuals to meet their nutrient needs within their energy requirements. However, we have found time and time again, individuals really like consuming added sugars at levels higher than the recommendations. Part of that is because sweetness is innately liked, and foods and beverages high in sugars are quite palatable. There have been some strategies proposed to help with reducing added sugar intake by the individual and at the population level. One such strategy is substituting reduced or no sugar added substitutes for full sugar products, such as swapping out your frosted shredded wheat for plain shredded wheat, swapping out yogurt with added sugars for yogurt with fruit, or substituting soda for sparkling water. Another approach is just reducing the portion or frequency at which foods with high levels of added sugar are consumed. So that would mean either reducing the size or how often you eat things like cookies, cakes and pies, sweet bakery products, or candy. So we have these strategies that all make sense to help individuals reduce their added sugar intake, but it's unclear how efficacious these strategies are for reducing added sugar intake by an individual and among the total population. Factors that influence efficacy of these strategies include factors such as how much sugar is the food that is being substituted for the high sugar product, and really how frequent are some of these high sugar foods even consumed. So before we can provide more strategies and recommendations for individuals to help reduce their added sugar intake, we wanted to take a closer look at the dietary patterns among individuals who are meeting and exceeding added sugar recommendations. So the objective of the results I'm about to present are results from a cross-sectional analysis to describe the dietary sources of added sugars and dietary patterns among adults using data collected as part of the What We Eat in America component of NHANES. In addition, we wanted to look at not only the sources of added sugar, but the dietary patterns among individuals meeting and exceeding that 10% of energy from added sugar recommendation. For this analysis, we used 2 cycles of NHANES, the 2015 to 16 and 2017 to 18 cycles. We used the What We Eat in America component of this to conduct our analysis, which is based on many of the data sets that Donna just described. And for this analysis, we focused on added sugar intake among adults that were not pregnant or lactating. So the 2 key data sets that we used were the Food Patterns Equivalent Database, which is FPED, as well as the Food and Nutrient Databases for Dietary Studies, FNDDS. So just to briefly describe how we conduct this analysis, we took the data collected from 2 cycles of NHANES and calculated the amount of added sugars from each of the foods and beverages reported by all adults in those 2 cycles. We then aggregated sources of added sugar consumed in combination with other foods and beverages, because we wanted to make sure that we were looking at how foods were consumed, given that sugars are commonly added to other food products. Food and beverages were then categorized into different categories based on how they were typically consumed. And then we calculated the amount of added sugar and the intake amount in grams of each of the food and beverage categories. We then further, in preliminary analyses, took that population and broke them into added sugar intakes groups based on the total amount of sugar they consumed in a day. We looked at individuals who are consuming less than 6% of energy from added sugar. That is the amount of added sugar modeled by the Dietary Guidelines Advisory Committee that would allow individuals to meet their nutrient intakes within their energy requirements. We also looked at people who consumed 6 to less than 10% of energy from added sugar. Those individuals are still meeting the dietary recommendations. And then we looked at incremental increases above that 10% mark. So individuals who consumed 10 to 14% of energy, 14 to 18%, and greater than 18% of energy from added sugars. I want to go into a bit of detail about how we developed our food and beverage categories. As Donna suggests, many foods consumed, as reported in NHANES, are consumed in combination. Approximately 40% of all foods are consumed as combinations. So this is particularly important for added sugars because they're typically added into other food products. So we took foods that included components of sugar in that sugar category that Donna described and looked at how they were added to other foods. Looked at how they're added to things such as coffee and teas, how they were incorporated into cereals, to take something from a low sugar cereal to high sugar cereal simply by adding the sugar. So in preliminary analyses, we found that about 7 grams of added sugar were consumed in these combinations, further highlighting the importance of looking at foods consumed as combinations as opposed to the individual FNDDS codes, particularly important when you're looking at added sugars. So after we aggregated all the combination codes that include sources of added sugar, we then regrouped these categories based on the What We Eat in America categories that Donna described. We took these and further grouped these into the categories that I have displayed here on the right. Just jump into some of our top line results. As we noted previously, added sugar intake is relatively high among the US population. Individuals are consuming on average about 13% of energy from added sugars, which equates to about 69.6 grams of added sugar, which is the mean intake of the adult population. So as I mentioned, we wanted to look at the intake of added sugar from our different categories, which I know is a bit overwhelming for this slide. So we're going to focus in on our top 10 sources of added sugar consumed by the US adult population. We sorted these categories here in decreasing order, and what you can see is that added sugar from sweetened beverages, including sugar-sweetened soft drinks, teas with additions, fruit, sport, and energy drinks, and coffee with additions, wrap up our top 4 categories of added sugar consumed by US adults. Combining these together equates to about 33 grams of added sugar, which is very high compared to the other categories. The remaining top 10 categories that contribute to added sugar intake that are contributing about 3.5 to 4 grams of added sugar include candy, ice cream and frozen desserts, cakes and pies, cookies and brownies. And then to wrap up the bottom of the list, contributing about 2.5 to 3 grams of added sugar per day include sandwiches and cereals. So as I mentioned, as part of our preliminary analysis, we wanted to look at how individuals who are meeting the dietary recommendations for added sugar intake are comparing to individuals that are exceeding intake. So you can see at the bottom of this graph, we have the total populations and our added sugar intake levels, the y-axis expressing the amount of added sugars from each of these added sugar intake levels by the different added sugar intake category. So for sugar sweetened beverages, teas with additions, fruit, sport, and energy drinks, and coffee with additions. As you can see, for these beverage categories, with increasing level of added sugar intake, the amount of added sugars from each of these food categories is increasing. For some of these categories, like the sugar sweetened sports drinks and the fruit, sport, and energy drinks, the added sugar level and the amount of each of these foods goes up with increasing level of added sugar intake. However, some of these categories, like teas with additions or coffee with additions, we see an increase in the amount of sugar within these categories, but the actual amount consumed is relatively consistent. This is suggesting that individuals are consuming the same amount of tea and coffee, they're just adding more added sugar when you're comparing individuals with increasing levels of added sugar. To look briefly at the other sources of added sugar in the diet, we see here the amount of added sugar with increasing level of total added sugar intake for candy, ice cream, frozen desserts, cakes and pies, cookies and brownies, sandwiches, and cereals. So the relationship is less consistent than as we saw in the beverages. So for many of these categories, the higher levels of added sugar intake tend to consume more added sugar from these categories, but the amount that they're consuming is not necessarily higher. Look at something like cereals. Many of these categories are consuming very similar levels. They're just consuming more added sugar at those higher levels of intake. What we found interesting was that individuals who meet the dietary recommendations and consume less than 10% of energy from added sugar don't necessarily abstain from consuming these foods that are typically considered to be high sugar. They are still consuming candy, ice cream, and frozen desserts, cakes and pies, and cookies and brownies. They're just consuming them at lower levels than those very high consumers of added sugar. So to summarize, added sugar intake among the US population exceeds the recommendation of less than 10% of energy from added sugars. Key contributors to added sugar among the total population are typically beverages, sweetened beverages, primarily those sugar-sweetened soft drinks. Candy and desserts, including ice cream, frozen desserts, cakes and pies, cookies and brownies, also contribute to added sugar intake, but they contribute quite a bit less than those beverage sources. We finally conclude with a recommendation for individuals who are exceeding added sugar recommendations. Targeting on reducing the amount of sugar from sugar-sweetened soft drinks is a good strategy to help individuals who are meeting the added sugar recommendation meet the added sugar recommendation. So what I presented for you now is just some preliminary analyses that we are still finalizing. The mean intakes of added sugar that are presented are representative of the general population, but we plan to look at usual intake of added sugar to get a better idea of added sugar intake patterns among the individuals who are consuming very low and very high amounts of added sugar. I want to thank everyone for joining us today for this presentation, and I specifically want to thank some of the collaborators that were involved in this project. Specifically, Lauren O'Connor and David Baer in the Food Components and Health Lab, and Alana Moshve and Donna Rhodes in the Food Surveys Research Group. And with that, I will let Kyle take it away for the rest of the presentation.
Fantastic. Thanks so much, Dr. Higgins. Our final presenter is Dr. Kyle McKillop, who is with the Methods and Applications of Food Composition Laboratory at ARS. Kyle's going to talk to us about added sugars and sugar data in Food Data Central. And it looks like you're ready to go, Kyle. Please.
Great. Thank you so much. So I'm going to bring us full circle back to Naomi introducing Food Data Central, and today going to talk about a breakdown of sugar data that exists within the system, and then just give you sort of a practical, how do you get to this data within our system? I'm going to show one example, and then I'll describe two other ways to get in and play with this data. Just a note first, I have nothing to disclose other than I'm a USDA employee. and that this presentation today was created using the October 2022 release of FDC, which as of today and the recording of this webinar is not available yet. It will be available by the end of this month, October. So first, sugars in the data set of Food Data Central. There is some caveats on to when you download the data, how sugars are described. So I wanted to start with explaining where and the two definitions of total sugars that exist within the data set, and then added sugars, as we are interested in today. The primary total sugar nutrient within FDC is called sugars total, including NLEA. And this is the historic mapping of total sugars within food composition data sets at the USDA. You're going to find this in branded foods representing the branded label total sugars, NLEA, of course, being the Nutrient Labeling Education Act. This is what we see in the survey foods, FNDDS, that Donna showed us earlier today, as well as the standard reference foods within the FDC data sets. Now, Foundation Foods handles total sugar differently. This was previously labeled total NLEA sugars, as when Foundation Foods was first released, all of the 3 monosaccharide and 3 disaccharide values were being analyzed and then summed together. That sum is what we call total sugars in Foundation Foods. Given that we do more targeted analysis on sugars, we've removed the total NLEA qualifier on those sugars and just call them total sugars. And then lastly, we have added sugars, and that only exists within the branded food data set. Added sugar values that we have here are as they're being reported on the nutrient facts panel and as reported by the data providers that are providing that data on that label data forward. So if you're interested in getting into this data, Food Data Central is fdc.nal.gov. If you go to the menu, you'll see next to food search is component search. You click on that, that's going to get you to a very simplified search that you can see here. Let me zoom in just a little bit here, and you can see that there is a drop down here for all of the components that exist within FDC. It is an autocomplete, which means if you start to type, in this case, I've typed out SUG, you can see the 3 sugars that I described earlier, and now you can anticipate where those sugars will come up. After I select one here, I'm going to select added sugars. I'm going to drop down to default sort method, and I'm going to choose here whether I want the results to be in a per 100 unit serving or a per serving size. It says per 100 unit. Unit in this case will be either per 100 gram or per 100 milliliter. So branded foods are allowed to be presented in per 100 milliliter servings, and so that's why the word unit here is used. After this, I'll select a per serving size. You then can search and get a set of results on the data set. Now, what's nice here is if you scroll down the page a little bit, it will tell you how many results are expected from that search. So in the case of branded foods, as of this next release, there are just over 120 branded foods that have reported some form of added sugar value forward in the data set. So after searching, the results look like this. All of the fields here are sortable by clicking on them. In this case, I've clicked on the added sugars per serving size and did the values in descending manner. And when I've done that, you might have noticed something if you are familiar with added sugars, which is the amount of sugar in this particular product. And this is something that exists through all components within branded foods. So I wanted to highlight it here and explain it as it's something you'll have to consider when using the branded food data set. And this example is stating that there's 930 grams per 411 milliliter serving, or that in other words, 226 grams per 100 milliliter serving. The data that we receive from branded foods is data as provided by the data provider. Efforts are done to communicate these obvious errors back to the data provider, but at this date, the only hard errors, meaning the only thing that blocks data from coming in, are data that's required and missing. Now, the partnership, the branded partnership that brings in this data and helps manage this data, are looking and evaluating more strict checks to be reviewed and to see if they should be implemented in 2023. But in the meantime, these records are being communicated back. And if you find a value or you want to know more about an error that you discover, you can always contact us. We can always communicate that message back to the data provider. Back to that search, again, at the bottom is a downloads or results. And so if you hit download results, it will download a CSV file or a comma separated values file that you can then bring into Excel. This is what it looks like when you bring it in. I want to highlight here what you can expect in some of the fields here. This has been standardized across all of the data types and meant to be a very quick way to bring in component searches through FDC. So it has a few caveats here that I want to highlight if you're going to use this data. The first is the FDC ID. So this FDC ID is a unique record identifier. Every unique product will have its own unique UPC or G10 identifier that's listed second here. So for all branded foods, the UPC G10 is that unique identifier, and you can see in column B that identifier is there. So if a product gets updated, the description changes, the ingredient list or the nutrients change, the UPC G10 identifier will stay the same, but the FDC ID, a new ID will be generated for that record. Another note here on the identifier for G10s is that if you just open this file from your computer, Excel may remove the preceding zeros from the identifier field. You can see that Excel has thought that this is in scientific notation, and in fact, the identifiers can have preceding zeros. The way getting around this is to open the file after opening Excel. So go through Excel, open the CSV file, and then make sure you identify that field as a text field, not as a number field, which will remove those preceding zeros. It doesn't change the value of the identifier. It's just a matter of if you're trying to map this to other data sets that have that preceding zero. You then have the value, which is a per 100 unit amount, and a measure value, which is the per serving size. And I've listed exactly how to calculate that, but here's another example. That first record there is saying the value is 13.2 grams per 100 gram, and that measure value is 5 grams per serving size, which in this case is 38 grams. Now the CSV has its limitations, so I also want to note that we have data source downloads. If you go to FDC and you go to the menu and you go to download data, it'll bring you to this URL where the files are available in CSV formats and JSON formats. And these files are generated every time we have an April and an October release. The biggest downside to these files is that Branded Foods is updated monthly, and it does not include the monthly updates of Branded Foods, only which data are available on releases in April and October. If you want to get to more recent data, we do have the FDC API, or Application Programming Interface. This is available by, again, going to the menu, going to API Guide, that will bring you to this following link. We always recommend to use our API. We know it requires some knowledge and maybe some training to use, but it's going to have better access to the full breadth of Food Data Central data, not only for branded foods, but for all our data types. It will have access to historic branded food data, not only the monthly updates, but also every previous iteration of branded foods is available and discoverable through the API. It will have better search functionality if you want to limit your search to specific words and to specific, for example, food categories and searching. And then it's provided in a structured data format, otherwise in this case known as JSON. You can see an example of this on the right. You can see it looks different than how an Excel file might work. An API is meant for you to take an application or a program that knows how to read from an API to have direct link and access to our database at any given time and pull the data that you're looking for. Again, for more information, go to that API guide where it goes into more depth about the API. And last, I just want to leave a note, which is if you want to contact us, if you go to Food Data Central's menu and go to Contact Food Data Central, it brings you to this URL. This form is great for not only asking us questions, but to giving us feedback and recommendations of features or clarifications on Food Data Central. We use this not only to try and give you an answer in a timely manner, but we also use this to gain feedback on how users are interacting with our application and the types of features that they were requesting as we plan forward into future releases of Food Data Central. And also my email. All right, and thank you very much. That concludes my portion today.
Great. Thanks so much, Kyle and everyone. If we could have the presenters come join us on camera, that would be great. And I'll put in a quick plug to please go ahead and type in some questions. Not surprisingly, we have lots of questions, however, already coming in. So if it's okay, I'm going to go ahead and start with the first question going to Dr. Dawson. And this first question actually asks, you talked about how there were tasters, and I know I've heard the term super tasters, right? Are tasters then more or less likely to like to eat cruciferous vegetables?
Well, that paper that was impregnated with PTC in your psychology class, if you were a non-taster, you more than likely don't taste the alloisothiocyanate that's in cruciferous vegetables.
Okay.
So that's a short answer, but you know, these receptors are promiscuous and they may bind other compounds, or other receptors may bind that compound. So I think the key is doing genotyping of an individual and then doing all the taste receptors and polymorphisms, and then coming along with your PTC and your test compounds to see whether they can test it or not, or taste it or not.
Got it. Great. Thanks. And then let's just kind of continue down the list here, and this one's for Donna. When you think about the reduction in sugar over time, right? You shared both total sugar and added sugar reducing in time. What do you think is the most likely explanation? Is it reformulations? Is it awareness from the consumer side? Is it diet sodas? What's your thoughts here on that?
Well, thanks. And that's a very interesting question. And I will give just, what my feelings are. And one, I don't think it's an increase in diet soft drinks, because at the same time period, consumption of diet soft drinks also declined. But and some of it is probably reformulation, but I think a lot of it is awareness. And about this time, there was all this talk about taxing sugar sweetened beverages, whether they actually were taxed or not, but I think that did increase awareness of it. So we are drinking less, and at the same time, serving sizes decreased. You could no longer buy, what was that super big gulp at some of the convenience stores. So that decreased. And also the consumption of tea increased at the same time, and the consumption of bottled water. So I think Americans are switching from some of these super sweet vegetables into ones that are less so. But, as Kelly showed, we are getting a lot of sugar from tea. So whether it's added, as we consume tea, or whether it's purchased as some of these sweetened tea beverages or sweet tea. So I think it's a combination of awareness and maybe some reformulations.
Great, thanks so
Just
much.
kind of a guess.
Please. Yep, nope, it's all good. Because one of the questions then is around, does tea with added sugar product category? I'm assuming that includes the ready to drink iced teas that you can buy. Yes, okay, all right, so that was one of the questions. So I'll move on to another question for you, Kelly. And it had to do with, and I'm gonna pose this to you, Kelly, but it's probably also kind of more broadly applicable. It looked like your analysis was really focused on the NHANES from 2015, 2018. We know that we had survey pauses because of COVID, right? So two components that I think would be interesting for the audience would be, could you talk, I think it'd be good for the audience to just understand the inherent lag delay between when the surveys get done and when they get released, right? And then two, any kind of reflections on how translatable data from pre-COVID is to data now.
Yeah, so I guess to get to the lag in data release, it's about a year or two between when the cycle is conducted and when the data is released. That's because they're collecting a large amount of data. They have to clean it, process it, make sure it makes sense before it's released, and then can be analyzed by other researchers. So there's a lag there. And then COVID. So during NHANES and COVID, dietary patterns shifted immensely. I believe did their best to capture data, on. But during COVID, individuals were eating out at restaurants in the beginning quite a bit less. And individuals consume a lot of sugar-sweetened beverages in restaurants when they go out to eat. You go to lunch, you get your diet soda from the fountain drink. So if I were to guess, I would guess that the amount of sugar may have gone down during COVID, just simply because you're not consuming those sugar-sweetened soft drinks that you would typically consume in a restaurant. So then how translatable are the results before COVID to during COVID to after COVID? That's a great question. I think we're still not entirely back to normal. I go into the office a couple times a week. I'm eating out at restaurants for lunch much less, and I believe other people could say the same. So I don't think, there are going to be some differences, and when that data is released, it would be really interesting to look at the pre-COVID life to post-COVID life. But I think that the pre-COVID life is more similar to now than COVID. So it's the best data we have right now, and the new cycles haven't been released. So stay tuned.
Great, thanks, Kelly. I want to bring in Kyle here for a question, and this has to do with, can FDC, can the branded foods database, can it be used to track, reductions over time, right? So that if that food formulator, right, as Donna was talking about, there's probably been an awareness change, probably a food formulation component to it, as the food formulations are changed, right? Is there an ability to kind of track that through the database?
Yes and no is the quick answer. So when a product goes through a small formulation change, then when they update that data on FDC, there will be sort of that historical record and that new record, and you can potentially see a difference there. More likely, that's probably more relevant for tracking a product before added sugar was required by labeling, and then looking to see how that value changed when added sugar was then required by labeling to see what was recorded. The biggest caveat here is that if a product is formulated enough, they're required to put a new product identifier on it. And a new product identifier also exists for every different packaging variation of the product. And so if it comes in a 2-pack versus a 10-pack versus a 200-pack, each one of those packs gets its own G10 and its own identifier. And there's been a lot of exploration on how to bring in common formulations. But the reality is if a product changes enough, it receives a new G10. So you're going to have to be clever in looking at brand names and product descriptions and sort of looking at products and historical products probably as groups or a whole, as compared to products that have been updated more recently with more recent data. So it is challenging, it will be challenging to play with that data to look at trends over time because of the way that those identifiers on products work.
Great, thanks, Kyle. Got a question here for Dr. Dawson, and this one, I'm going to do my best, is asking about polymorphisms, allele variations, and how there was some variations where you made a comment around specific polymorphisms, allele variations being tied perhaps to Asians versus African Americans, and the HbA1c levels and all that. Could you just expand a little bit more around that around that topic?
Sure, that was in the context of the taste receptor 1 type 2. And
Okay.
there were two, we'll call it two unrelated pieces of data. One was, if you look in the GenBank at the SNP database at that particular variant, it gives you results from the 1,000 Genomes Project, which looked at various individuals around the world. And if you take a look at that average, it's 31% for the presence of the codon for valine, not the valine or isoleucine, but just simply the presence of the DNA sequence for the valine coding regions. That was 31%. And then they averaged, they had Asian breakdown, and then they had East Asian breakdown and the like, but that was around 22%. And if you go to Africa, it was around 40%. Now, if you talk about hemoglobin A1c, that was the relationship between that particular polymorphism and people who were not necessarily pre-diabetic, but it correlated with their overall hemoglobin A1c level. And that was a paper that was put out just this year.
Okay.
Hopefully that answered it.
Yep. No, great. Thank you very much, Dr. Dawson. So looking at a couple more questions here, and I'm checking the time. Yep, we've got time for it. You know, one of the questions is, and this is going to, I'm going to pose it to you, Kelly, but it may be for others as well. You know, in the NHANES analysis and that you showed, is there a way to consider dietary quality impacts at the different levels of sources on the added sugar intakes? I can try and translate that further. But
yeah, it would be possible to look at diet quality using something like the healthy eating index among individuals who consume different levels of added sugar. Some of the future projects that we're going to be working on would be looking at usual intake of added sugar as a percent of energy. So we plan to be looking at the different food categories, which, you know, intake of those categories is one way of looking at dietary patterns, but seeing how diet quality varies would be something that would be interesting to look at, and something we haven't considered, but would be a good idea. So yeah, Donna, I don't know if you have, you know, the FPED database that Donna discussed is really set up nicely to help incorporate that into measures of diet quality, looking at using the healthy eating index.
Yes, and I agree. It could be done.
Please.
Yes.
okay, it could be done. That's perfect.
It could be done. It's the HEI scores tend to be low for most Americans, but there might be a difference between those in, you know, Kelly's groups of the percent of added sugar.
Okay, terrific. Terrific. Thank you for that. So we've got two questions coming in. I'm going to make these the final two questions. One, which is one that has been asked a couple times, is when can we expect the next NHANES data release?
Well, I'll take a stab at that. And
Perfect.
when the pandemic hit, it really curtailed collection of data. And there has been a data release, and it's termed 2017 to March 2020 pre-pandemic. And that has been released by NHANES. And you can get it from our website or from the NHANES website. And we have released the food categories to go along with that combined set.
Okay.
But that's the last set. There was not enough data to be nationally representative to release a 1920 data set. And currently, NHANES is back in the field and they are collecting data. Right now, they started sometime in 21, and hopefully there'll be a release of 21-22. When that comes out will really depend on when they stop their collection. To get enough sample, they will probably continue on until 23. So no data will come out until they have completely, finished their cycle. So I wouldn't expect that until sometime in 23. And we don't know what that sample will look like because there have been some challenges just dealing with data collection post-pandemic. So we just have to wait and see what happens. But unfortunately, it, will not be released until probably 2 years from now.
Got it. Got it. Nope, understand. And then other question here, and this will be the last one, is, as a team, you all provide, I'm going to sit and say the government basis, right? Which I know gives many of us great comfort with intake trends, right? Food intake trends. And question is around, you can look at whether it's ANOVA, Mintel, Hartman, Nielsen, and others. How closely do you guys take a look at that stuff? This is just out of curiosity. You're allowed to say you're aware of it and it comes up occasionally, or you can say, yeah, we look at it with great interest. I guess it's kind of that.
Well, we have looked at it for just seeing what the highest, what's most, I don't know, purchased in the US, but with our database, we only have 8,000 codes, and now we even have less. So it nowhere captures the amount of data that is in branded foods or is in some of these other
Right.
databases. So we do look at it, and we tend to do more generic, a generic representation of foods rather than specific foods that you would find in those databases.
Great. Thanks so much,
Because a lot of
Donna.
people, a lot of participants don't really know the details of the food. They don't know what they ate. They can't give you that. And we're also collecting data for a 2-year period, and it needs to be matched really to the same database. So that's
Right.
another challenge.
Right, right. No, as IFNS is a proud partner on the Global Branded Foods Database, I have a working level of astonishment on what goes in on the data quality aspects of it. So I can only imagine when you start bringing in the intake component. So thank you all. And with that, let me click here. Let me start by thanking all of our presenters here today. Want to just quickly note that for those of you who are registered dietitians, there'll be an email that comes out, and you'll need to complete that survey to get CEU credits. Also want to thank everybody for their time this afternoon. I know I learned a great deal, and based on the questions and the interest, I think our audience did as well. We have our next webinar coming up next month on the complexity of carbohydrates, which should be fascinating. As we go to wrap up, I will do a real quick plug for all of our attendees that we have opened up our call for abstracts for the IFNS Science Innovation Showcase. Our Innovation Showcase is a virtual event held December 13th to 15th. But if you've got some science that you want to share, please do visit our website and know that abstracts are due by November 14th. With that, thank you everyone for your time and attention today. At IFNS, we look to connect, collaborate, and catalyze science that matters. Please check out our website at ifns.org, follow us on Twitter, and follow us on LinkedIn. Those three mechanisms are the best way that you can stay informed on how we are delivering science that matters. And with that, and with great gratitude to the Beltsville team, thank you everyone, and have a good rest of your day. Bye.